Life's diversity, from the deepest ocean trenches to the highest mountain peaks, showcases nature's incredible variety. It's a breathtaking display of adaptation and resilience, a testament to the power of biological processes at play. But beneath the surface, this vibrant world is built on a microscopic marvel, the protein. These tiny, intricate molecules are the workhorses of life. Proteins are the engines, the driving force behind nearly every biological process. They act as messengers relaying crucial information between cells and tissues, and they function as defenders within every cell, protecting against invaders and maintaining cellular integrity. They are constantly performing countless vital tasks, ensuring the smooth operation of our bodies. They provide structure, the very scaffolding upon which our bodies are built. Our hair, skin, and nails are primarily made of them, showcasing their role in physical appearance and protection. And they act as enzymes, biological catalysts that speeding up life's essential reactions, making them happen in a fraction of the time they would otherwise take. Without proteins, our bodies would simply not function. Life as we know it would be impossible. Their versatility comes from their unique three-dimensional shapes, a characteristic that dictates their specific function. Each folding precisely to perform a specific job, like a key fitting perfectly into a lock. The instructions for building every protein, the complete recipe for life's molecular machines, are stored in DNA, our genetic blueprint, the master code that governs our existence. This blueprint is read and translated into proteins, a complex process of decoding and construction, through a process called protein synthesis, a remarkable feat of cellular engineering. It's a journey from genetic code to functional machine, a transformation that underpins all biological activity, happening billions of times each second in every cell. It's a constant flurry of activity, a testament to the efficiency of life. Understanding this process, unraveling the mysteries of protein synthesis, reveals how life's complexity is built from simple instructions, how order emerges from apparent chaos. This is the story of protein synthesis, a tale of molecular machines and the genetic code that brings them to life. Deep inside each cell's nucleus lies DNA, the master plan for life. DNA's code is written in 4 bases: adenine, guanine, cytosine, and thymine, arranged in a precise sequence. Segments of DNA called genes each contain instructions for building a specific protein. The cell reads these instructions using 3-letter words or codons, each coding for an amino acid. Even a single mistake in this sequence can change a protein's shape and function. To build a protein, the cell makes a temporary RNA copy of the needed gene. This RNA copy can leave the nucleus, carrying the instructions to the cell's protein factories. The first step in this process is called transcription. It's how the genetic code is set in motion. Transcription, a fundamental process in molecular biology, begins in the nucleus. This is where the magic happens, where the genetic code starts its journey to becoming a functional protein. Inside this command center, RNA polymerase, a crucial enzyme, locates a gene's specific starting point on the DNA strand. It's like finding the perfect address on a map. Ensuring the right gene is transcribed, the enzyme then meticulously unwinds the DNA double helix, separating the 2 strands to expose the genetic information. This unwinding is essential for accessing the code. With the DNA unwound, RNA polymerase carefully reads one strand, acting as a molecular scribe. It's like reading a recipe to prepare a specific dish. Using this strand as a template, it begins building a complementary messenger RNA molecule, often referred to as mRNA. This is the first step in creating a portable copy of the genetic instructions. This newly synthesized mRNA will carry the genetic code out of the nucleus. In this process, RNA uses uracil instead of thymine as one of its nucleotide bases, a key difference between RNA and DNA, but otherwise the base pairing rules remain the same. Adenine pairs with uracil and guanine pairs with cytosine. This ensures the mRNA is a faithful representation of the DNA sequence. As RNA polymerase continues to move along the gene, it elongates the mRNA molecule, adding nucleotides one by one. The result is a faithful mRNA copy of the gene, ready to be translated into a protein. When RNA polymerase reaches the end of the gene sequence, a signal tells it to stop. At this point, the enzyme releases both the original DNA template and the newly synthesized mRNA molecule, setting them free to perform their respective functions. The DNA double helix then rewinds back to its original form, completely unchanged by the transcription process. Meanwhile, the mRNA carries the genetic message, ready to be transported out of the nucleus. This mRNA is essentially the blueprint, containing all the instructions needed for building a specific protein, the workhorse of the cell. Transcription is a vital protective mechanism, ensuring the original DNA stays safe and protected within the confines of the nucleus. The mRNA, now complete with its genetic instructions, is ready to move on to the next crucial step in gene expression, translation. It will soon leave the nucleus through nuclear pores, carefully carrying its precious instructions to the ribosomes, the protein synthesis machinery. The journey continues from the nucleus to the ribosomes, where the genetic code will finally be translated into a functional protein. Before leaving the nucleus, the mRNA undergoes editing. Non-coding regions called introns are removed and the coding exons are spliced together. Protective caps are added to each end, stabilizing the molecule. The mature mRNA exits the nucleus through nuclear pores, entering the cytoplasm. Here it's no longer protected, it's in the cell's busy workshop. The mRNA's next destination is the ribosome, the cell's protein factory. The message is ready to be translated into a protein. The journey from DNA to messenger is complete. Now the real construction begins. Deep within the cell's cytoplasm, a critical process is about to unfold. The messenger RNA, or mRNA, carrying the genetic code, embarks on a journey to find its designated protein synthesis factory, the ribosome. The ribosome, a complex molecular machine, recognizes and clamps onto the mRNA strand, much like a reader carefully positioning themselves on a tape, ready to decode the information it holds. This binding is the first crucial step in translating the genetic message. The ribosome then meticulously scans the mRNA sequence, searching for a specific signal, the start codon, AUG. This codon acts as the initiation point for protein synthesis, marking the precise location where the ribosome should begin its work. The AUG codon sends a clear signal, indicating exactly where the ribosome needs to position itself on the mRNA to begin the translation process. It's like a molecular GPS, guiding the ribosome to the correct starting point. Interestingly, this AUG codon not only signals the start of translation, but also codes for the amino acid methionine. Methionine is often the first amino acid incorporated into most newly synthesized proteins, acting as the initiator of the polypeptide chain. Now, a special molecule called transfer RNA, or tRNA, comes into play. Each tRNA is specifically designed to carry a particular amino acid. In this case, a tRNA carrying methionine approaches the ribosome. The tRNA recognizes the AUG codon on the mRNA through a complementary sequence called an anticodon, ensuring that the correct amino acid is delivered to the ribosome. The ribosome meticulously ensures that the mRNA is read in the correct reading frame, preventing any errors in the amino acid sequence. This precise reading frame is crucial for ensuring that the protein is built accurately with the correct sequence of amino acids. With the first amino acid, methionine, now correctly positioned within the ribosome is primed and ready to continue the elongation phase of protein synthesis. A steady stream of tRNAs will now begin to arrive, each carrying its specific amino acid. Each tRNA will match its anticodon to the next codon on the mRNA, ensuring the correct amino acid is added to the growing chain. The molecular assembly line is now fully operational, ready to churn out a new protein. And as each amino acid is added, the protein chain begins to grow longer and longer. Translation has officially started, marking the beginning of a complex and vital process. The ribosome continues to read the mRNA sequence, and the tRNAs diligently deliver their amino acid cargo. As the chain grows, it begins to fold and twist, eventually taking on its unique three-dimensional structure. The entire process is a testament to the precision and efficiency of cellular machinery, ensuring the accurate production of the proteins that drive life. The ribosome moves along the mRNA, reading one codon at a time. Each codon attracts a matching tRNA carrying a specific amino acid. The ribosome links these amino acids together, forming a growing chain. After delivering its cargo, each tRNA is released to be reused. This cycle repeats rapidly, building the protein step by step. When the ribosome encounters a stop codon, the process halts. Release factors help disassemble the complex. The completed polypeptide chain is released into the cytoplasm. Translation is finished, the protein is born. The cell is ready for the next task. The new polypeptide chain must fold into a precise three-dimensional shape to function. Sometimes helper proteins called chaperones assist in this folding. Once folded, the protein travels to its destination, inside or outside the cell. Proteins power every aspect of life: digestion, movement, immunity, and structure. This process, from DNA to functional protein, happens constantly in every cell. It's how our genetic code becomes reality. The silent ballet of protein synthesis sustains life, moment by moment. Every heartbeat, breath, and thought depends on this microscopic marvel.